Thanks for coming to the second computational biology seminar for the fall 2014 semester. Our speaker tonight is our very own Brant Faircloth. Brant is a new faculty member in the Department of Biological Sciences here. He's originally from Georgia, did his undergrad and graduate work at the University of Georgia, worked in the South Carolina Department of Natural Resources for a while, moved on to UCLA for a couple of postdocs, and then joined us here just, I guess it's been 2 months ago. So he has done some really seminal work on how we gather genomic data to try and answer interesting questions about evolution. And in the process uses a lot of computational techniques to try and process all that data. So he's going to, I think, talk about his history of biological interests from the field to the computer.
Okay, so I don't know exactly what you all do. How many of you have a whole lot of experience doing computational work, whether it be programming or just using computers? Just raise your hand. Okay, so one person. How many of you want to do a fair amount of computational work? Okay, 5 people, 6 people. How many people don't really care that much about computational work? It's okay if you raise your hand. You're not going to lose any points. Okay, so I won't even say the number, but a few people, right? So lots of my work is computational. I'm actually going to give a talk that's a little bit different than my normal science talks. This is really sort of an informal talk about how I went from being essentially a field biologist to being a computational biologist, what people call a computational biologist. So I have to figure out how to make this thing work. So my name's really easy to remember. I'm named after a goose. There's a Brant goose. I don't know if you've ever seen these. It's Branta is the genus that they live in, or that they are in. And basically, when I was born with sort of this inherent love for nature. And like Jeremy said, I went to the University of Georgia. I got a degree in forestry and natural resources, which most people think means that you're going to be a park ranger. But what I did when I graduated, when I worked for South Carolina, was that I worked on red-cockaded woodpeckers, and I worked in South Carolina. I also worked for a while in Florida. And one of the things that I did was basically band the nestlings, which you see on the right here, and put in artificial cavities for the adult birds, which are on the left. So these birds, if you know nothing about them, are cavity nesting woodpeckers. They're an endangered species in the southeast. They used to be native to the longleaf pine wiregrass ecosystem that was all over the southeast. But basically habitat loss and encroachment of people on these longleaf pine forests really reduced their numbers. So while I was working on red-cockaded woodpeckers, I absolutely fell in love with this longleaf pine wiregrass ecosystem, which generally looks like this throughout most of the country. And when I started graduate school, I wanted to work in the same ecosystem. So I accepted an offer to work on a project on bobwhite quail. Any of you guys seen these before in the wild? No? No one? One person? Okay. So this is a northern bobwhite. This is a male bird. Males have white on their head, and they have this black eye line. I'll show you the female in just a minute. The chicks look like this. This is my obligatory cute baby picture. This is a one-day-old bobwhite. And unlike a lot of songbirds, these guys are born fully feathered. So when they hatch out, they're ready to go. If you know anything about avian biology, this picture is a really nice example of the egg tooth on birds when they're born that they use to get out of the egg. And this little egg tooth falls off after about 24 hours. As these guys get a little bit bigger, this is about a 2-week-old juvenile northern bobwhite. They look pretty much like this. That bird is probably about the size of your fingers. And we would band all of the juvenile birds with these little wing bands that we would find later when we caught them, provided that they lived. So my project was basically to understand where bobwhite babies come from. And these are basically the same age chicks as you saw in the image before. This is a female northern bobwhite, and she has to keep her babies warm, so she keeps them sort of tucked up in her armpits while they're out during the day and it's cool outside. And really what I mean by where do these babies come from, obviously we all know that. But I was interested in studying what the mating system of bobwhites was. So are females monogamous? Are males monogamous? Are these birds polygynous? Are they polyandrous? Et cetera, et cetera. And to do that, one of the things that I did is I would collect feathers from adult birds in the field, and we would use these feathers for genetic analysis. So if you've done any lab work in the past, what we were doing at the time, which was really cutting edge and isn't any longer, was microsatellite genotyping the DNA that we extracted from these little feathers right here. We'd take 10 feathers, we'd extract DNA, and we'd genotype them. We would also radio collar every bird in the field. So this brown thing that you see down here is basically a necklace radio transmitter. It goes over the head, you put it under the feathers. It lasts for about 6 to 9 months in the field, and it basically just emits a beep every 20 seconds. Once we put the radio collars on, we would let the birds go in the field, and we would keep them in this little box overnight and just let them out in the field in the morning. And then we would track them around. This is basically an antenna that's meant for tracking the signal that comes from radio collars, and it attaches to this receiver. And basically, wherever you point the antenna, if you're pointing towards the transmitter, the beep gets louder. If you're farther away, the beep gets quieter. And this is long ago, and it had much more hair. Now, one of the things that we would do, because we could find all of these birds, when females or males, so males incubate nests in the species, which is a little bit different. Whenever they're incubating a nest, they basically don't move for most of the day. So one of the things we were able to do, because we had transmitters on them, is that when we were in the field and these birds weren't moving around, we basically assumed that they were incubating a nest. And this is the nest right here. They normally have somewhere between 14 and 16 eggs on average, but that number can vary really widely. It can be from 4 to 28. When the eggs hatch, the babies basically run off with whichever parent is taking care of them, and they sort of putter around the woods and they eat insects. And one of the things that we could do is if we went out really early in the morning, this is about 5:45, but we had to go out about 3:45 every morning, we would find the radio collared mother or father with the offspring. We could build this fence around them in the dark, and then when the sun came up, we pulled up all the vegetation, and the parents flew to the other side of the fence and the babies ran around and we picked them up. And what we ended up with is just sort of this little bag of baby quail. Once we had the bag of baby quail, we would collect tissue samples from them, and we would also mark them on a chin with a Sharpie. It's really high tech. And we would use different colored dots to indicate which individual was which. And these tissue samples we would also use for genetic analysis. So we sampled the parents, we sampled the offspring. We can use genotypes to figure out which offspring go with which parents. So over about 8 years of grad school, which is a long time, I hired about 4 technicians. We captured about 1,380 adult birds. We took about 48,000 telemetry locations over 3 years in the field. That's about 12,000 per person, which is a huge number. I took about 24,000 on my own, and my boss, sort of true to form, took 15 over that period of time. When we genotyped all these guys, if you've done any microsat genotyping, has anybody in here done any molecular lab work? No? A couple? Okay. So the genotypes here, we genotyped everybody at 16 microsatellite loci, which again at the time was a whole lot, and now it's really not that many. So where things started to get really hairy for what I was trying to do is after I collected all these data, I had sort of adult captures when we caught the parents and we kept a record of when we caught them. I had all these telemetry records. We also, when we were doing telemetry, we would record any other birds that were near the bird that we were looking for because they all had transmitters on and you could usually pick up other transmitters. I also had all of these juvenile capture records. I had all of these genotypes. And then I had parentage inference data where we used the genotypes from the parents and the offspring to figure out which babies went with which parents. And the problem that I ran into is that I had to take all of this stuff and mash it together to try and get to some understanding of the system, right? Which parents are responsible for which offspring, and what is the mating system of those parents, right? Are some females producing offspring with more than one male, or are they always just producing offspring with one male? And what I basically needed to do was to store these data somehow. And back in the day, not terribly long ago, something like Excel was limited to only 65,000 rows of data. So you couldn't use Excel to store the amount of data that I'd collected. Not only did I need to do that, I needed some way to query the data. So across all of these different tables, maybe I wanted to know where a particular female was on a particular day, which chick we captured with that female, which female and male belonged to that chick or vice versa, and then what our parentage results said. So I had to keep the data and had to put them together in different ways. The other thing that I needed to do was to analyze the data. A lot of times the format that you collect them in is not the format that you need to analyze them in, right? It's just sort of the way that big data collection goes, or any data collection goes. You always find that you need to transform the data that you've collected. Similarly, sometimes you need to transpose the data that you've collected and basically go from columns to rows, from or to rows and columns. And finally, I needed to analyze the data somehow. So I needed to run statistical analysis on these. And what I found when I started to do this was that I had no idea how to do all of these things. And every component that I looked at was just sort of a mishmash of stuff that didn't make any sense, right? I had no idea what the SQL language was or what a database that used SQL was for. I had no knowledge of R. I knew that these things sort of drives this Linux operating system. And then I knew I needed to do some programming, but I didn't know how to do it. And I knew that I needed to type into something, and I didn't know how to make that work. So basically, I was just totally lost. I kind of knew that some of these pieces went together, but I had no idea how. And essentially, it was just like the Tower of Babel, right? It was all of these languages, and I understood none of them, but I knew that they might be useful to me somehow. And, it kind of boils down to just sitting there and not knowing what's going on. And if you've done any of this sort of thing, you kind of maybe have bumped into that. Or if you're taking a programming class, you've probably run into a situation where you've tried to make the computer do something and you cannot figure out how to do it. And it's frustrating, super frustrating. And so I thought, well, I'll just buy some books and I'll read the books and then I'll make everything better. And the books help, but they don't really help that much, because the trap that people fall into is they buy a book, they read the book, they don't really do the exercises, just like doing homework. You read the questions and you're like, oh, that's pretty easy, and you skip it. And I started to really move beyond the book, or basically just do what the book said to do, and work with these simple programming examples. So that's a little bit light, but the beginning thing that you do in every programming language is just to make the programming language print hello world to the screen. And as I did that more and more, I started to get a little bit better. I figured out how to uppercase words and lowercase them and reverse them, and use different data structures to do different things. At the same time, I started to dig around and figure out what the command line was like. Does anybody even know where the command line is on your computer? How many of you use a Mac? Okay, so a bunch of you use a Mac. Do you know what the terminal is? Or when asked if you knew what you were doing. So nobody knows even really what that does, right? So the terminal is an old school way of interacting with your computer, and instead of using a mouse and a GUI, the graphical interface, you basically type commands in. And for a lot of things, it can be much more efficient than doing it with a mouse. Okay? And for big machines like the supercomputers here, basically the only way that you interact with them is through this command line interface. Okay, there's no clicking, there's no mouse on the supercomputer. I learned how to compile programs, which is crazy in and of itself, and a lot of it doesn't make any sense. You just sort of follow the instructions and hope that it works. And one of the things that I started to do was really break things, right? And sometimes I broke them in a bad way. I broke web servers. I broke computers. I basically inadvertently erased an entire hard drive on the computer that I was using. Don't want to do that. And after I'd done all of this, it was just extremely frustrating, right? Like, all of this was frustrating, and I figured that I would never, ever figure this out, right? And it's kind of like the scene from Office Space where they go and they just beat the crap out of the printer, right? I mean, I just could not figure this out. Could not. And if you haven't seen this movie, you should watch. But over time, and after going through all these really, really frustrating steps, I started to kind of get a little bit of an understanding of what I was doing. And I started to piece little things together that became more helpful to me. So in this example, basically I learned how to count the number of sequences in a file, which doesn't sound like something hard to do, but if the file that you're counting sequences is 40 gigabytes long, it's a reasonably challenging thing. And I started to mess around with my programming, and I evolved from basically just having the computer print hello world to the screen to actually writing functional programs, right, that did things that I needed to do, that did this transposition of data, and that did analyses on my data. And in 2008, one of the things that I decided that I was going to do is I had a problem. I worked with microsatellites, and finding them and designing primers for them was a really challenging thing. So I actually wrote a program to make my life easier and do a lot of this work for me. And luckily, I was able to write it up and get it published. And to this day, this is still my most cited publication. And I wrote it in 30 minutes. So if you're interested in doing this type of work, I guess one thing that I would say is that progress comes really, really incrementally. It doesn't seem like you're getting anywhere. It seems like you're running into walls constantly. The increments seem really, really small, but you are actually getting somewhere. And with practice and a lot of struggle, you can and will get better at doing these types of things. So if you're trying to learn this stuff now, don't be completely frustrated, but try and work through the problem that you have. And one of the things that I did that really helps, and people ask a lot of times, well, how did you start to learn how to do this stuff, is I started working on my own problems, right? I didn't exactly do the problems that were given to me in the book because they didn't make sense to me. I started working on things that made sense to me and that made my life easier eventually. So now what I'm going to do is talk a little bit about how I transitioned from this really frustrated point to being able to use computation as sort of the basic unit of my workday. When I finished graduate school, I went to UCLA and I was hired actually to work on a project having to do with Drosophila melanogaster. You can say it different ways depending on what part of the country you're from. It's the common fruit fly. You've probably all seen them around here in labs or on your food. They're probably in your house. And what I was hired to do is basically repeat this study by A.J. Bateman from 1948 about male versus female reproductive variance. Okay, so this is a really important paper. People who work in the animal behavior realm cite it all the time. And what we were basically doing was repeating the experiment and making sure that we got the same results. Now, the problem with that experiment is that the whole thing went belly up. I was hired to do this. That was my job. And I found out that part of it had been so screwed up by someone else that I couldn't do what I was hired to do. And all of us were extremely sad, right? I mean, this was like 3 years of lab work in another lab that basically we threw in the trash. And what I had to do was figure out what I was going to do now. I was hired, I had a job, but I had nothing to do. And I kind of returned to my roots a little bit and started thinking about species. And one of the things that's interesting about bobwhites is that they're between 18 and 22 subspecies in their range, which is from America down into Mexico. And one of the things that we know about bobwhites and other species is that they're really diverse. And one of the things that I've been interested in for a long time is really how does this diversity arise, right? How does history shape the diversity of these taxa? Or if you look at it at a more broad scale, why is it that we have the bird species that we do, and how are they related to each other? And one of the things that we've wanted to do for a long time and we haven't been able to do is not only to understand how all these species are related to each other, but also understand how those species level effects translate to populations of individuals of a species and actual individuals of the species. And if we can figure that out, one of the things that we can do is we can explain exactly why species are so diverse. Why do we see this diversity of species that we do, whether it be fish or birds or snakes or mammals or anything? And one of the reasons that this has been really hard over time is that we've been able to work pretty well at the species level, and we've been able to work pretty well at the population level, and we've been able to work pretty well at the individual level. But what we haven't been able to do is to work across all of these levels in a reasonable way. And if you've done any molecular work, you might know that for species level questions, we tend to use slowly evolving markers, so parts of the genome that change really slowly. For population level questions, we use parts of the genome that evolve a little bit faster. And then for individual level questions, we use parts of the genome that evolve even faster. But those parts of the genome that we're using aren't usually compatible with each other. And so one of the things that I thought that I could work on was to come up with a type of genetic marker or a part of the genome that would accomplish all of these things, right? And it's sort of the holy grail of collecting data for studying these types of questions. If you can come up with something that does this, you've really come up with something useful to a lot of people and that will allow you to answer a lot of questions. And so one of some of the criteria for this type of molecular marker is that it's universal across taxonomic groups. So you can find it in snakes, and you can find it in lizards, and you can find it in squirrels, right? And that it's informative at different times so that we can use it for species questions, we can use it for population questions, and we can use it for individual questions. And that it's abundant, right? We want lots of these things in the genome. And finally, that we can come up with a way to collect the data. And so in 2001, 2002, and 2004, the human, mouse, and chicken genomes were published. And one of the things that I thought we might be able to do is to use these genome sequences to answer questions in other taxa that aren't humans or mice or chickens. And luckily, I came across this paper by a guy named Gil Bejarano, who works at Stanford now, a very nice man, where they basically did genome-genome alignments of mice and rats and humans. And so a genome-genome alignment is simply where you take the entire genome sequence of one of these organisms and you align it, you line up the similar pieces to the other organisms that you're aligning it with. So what they did is they just took the human genome and they tried to find the parts of the rat genome that were similar and aligned to the human genome, and they did the same thing for mice. And when they did that, what they found were these pieces of the genome that were really, really, really conserved. There were about 481 of them in the genome. They were 100% conserved between humans, mice, and rats, right? So very divergent organisms, very different critters. But they had these 500 segments that were completely identical over about 200 base pairs, okay? And the other thing that they found is that these units of the genome were only found once within the genome. So you didn't find the same thing in two places. And what I wondered was, well, can we use these really conserved parts of the genome as this molecular marker? We know that they're really conserved, right? We found them in these really divergent taxa. And what I thought is, hey, if they're really conserved, we can go in and we can find them in snakes and lizards and squirrels and bats. And the other thing that I wondered is the center of these regions may be really conserved, but as we move out from the parts that are really conserved, do we see that conservation go down, right? So do we see them become more different in different species? And if so, they may be useful at shallower times, right? So the species level is really deep in time, population level is less deep in time, and individual level is pretty shallow in time. So one of the things that, or some of the things that I needed to figure out were, are these UCEs conserved across amniotes, right? So we know that this other paper found them in humans and mice and rats. But can we find the same thing in alligators and turtles and bobwhites? And this is a paramyscus, so it's like a mouse, but not the same as a lab mouse. The second thing that I needed to figure out was, do they record evolutionary history? So can we use them to say something about how individual organisms are related to each other? So the first thing that I did was that I decided that I was going to do my own genome-genome alignment. And that I, instead of using human, mouse, and rat, would use anolis, right? Zebrafinch, and chicken, which were three other genomes that were available at the time. And kind of like with the learning how to use the computer situation, I was confronted with this just total bombshell of different programs and different data formats that I knew nothing about, right? I had no idea what any of these things were when I started to do this. And basically what I had to do was sit down, read about them, play around with each of them, and figure out how they work so that I could figure out if they would be useful to me in my work. And after I sort of picked a subset of these tools to work with, I was able to use my sort of budding coding skills to align these genomes and to inspect the alignments for regions that were really highly conserved. And when I did that, what I found was really cool, is that across these genomes, not only were there a number of UCEs, there were about 5,600 of these loci that were 60 base pairs long, so a little bit shorter than what they found in the original paper, and that they were 98 to 100% conserved, so a little bit less conserved than in the original paper. And so the next thing that I did is I wrote some more computer code to take all of the regions that I had found and try and find them across all of the other genomes that were available at the time. So human, chimp, rat, horse, cow, hedgehog, elephant, possum, platypus, right? And a whole list of other genomes that were out. And when I did that, I was basically able to find that with a few exceptions, these loci were present in all of these very, very different organisms. So it basically showed that these loci are present in amniotes. Now, the second question was a little bit more complicated. What I wanted to know was not only are they present in these organisms, but when we use them to infer the evolutionary history, the phylogeny of these taxa, do they actually give us a reasonable answer, right? Do we see that humans are more closely related to apes than they are to turtles? If they were more closely related to turtles, things would be broken and it wouldn't be a really useful thing. And so basically what I did was take all of the data that I'd found in these different taxa, I aligned them all together, and I inferred, using a couple of computer programs, the phylogeny from these data. And when I did this, you don't have to study this tree, but the bottom line is that what we found out was that they recover the relationship between the taxa that we expect. Okay, so we see that humans are more closely related to apes than they are to turtles. So the next thing that I had to figure out is, well, okay, this is great. I've done all of this in the computer, but can I actually take what I've learned in the computer doing this sort of genome-based, almost simulation work, and transfer that knowledge to an actual system with real organisms that we don't know anything about? And one of the things that I didn't know, or that I knew, was if you've done any lab work, PCR is really challenging, right? It's hard to get it to work. When you're working in different taxa, it's really hard to get it to work. And when you're trying to collect the number of loci that I found, it's tons of work. So if you worked for 33 days in the lab, you would have to do that constantly and have everything work 100% to collect all the data that I need. The other problem that I had was that if you have done any PCR, it requires that you usually design primers on each side of the thing that you want to amplify. And really what I was trying to do was completely the opposite of that. We knew that this part of the genome was conserved in the middle, and the ends were variable. So if we design primers for that region, it won't work. And what I wanted to do was come up with a new way, or find a new way, to collect these data just by grabbing the center piece of DNA and pulling it out of the background of all the other DNA. And I got really lucky at the time. There was a technique invented that was called sequence capture, and it sounds exactly like what it is. You have some regions in the genome that you want. You design some, they're called probes or baits, that are very similar to the region that you want. You stick the baits in with your genome. You hybridize them together. You stick them together with temperature. And then you actually just pull out the region that you want, and you leave all the green stuff that you don't want behind, right? So it's like fishing. You're just pulling out the parts of the genome that you want. And the really neat thing is that this technique, if PCR is down here in terms of efficiency, you can reasonably amplify about 10 loci, or 10 parts of the genome with PCR. With this other technique, you can pretty easily capture about 10,000 to 1 million targets in the genome really easily. So it's much, much more efficient in terms of the data we can collect. Now, the problem with this is that I didn't know how to use it, right? I didn't, again, like figuring out how to program or figuring out how to use Linux or figuring out how to do genome alignments. I didn't know how any of this probe stuff worked. It was brand new, and a lot of people didn't know the details. And so I actually wrote a fair amount of computer code to try and answer some of these questions. How do we design the probes? How many probes do we need for each region we want to capture? Can we enrich stuff from more than one sample at a time? And basically, the other thing that I needed to do was figure out a test that would be a reasonable test of whether or not this worked. And the test that I wanted to use was to try and reconstruct the lineages of ancient birds using data that I collected from these loci. And one of the reasons that we picked ancient birds is that no one on Earth really disagrees about these major relationships. And so we figured that if we could reconstruct those, we wouldn't have a disagreement on our hands and everything would be good to go. So I finally figured out which probes I wanted. I ordered them from a company. Basically, it's like ordering something from Amazon. They send you these probes. You mix them with the DNA that you want. They bind to what you want. And then you wash away the stuff that you don't want, and you keep only what you do want. And one of the things that we ran into, another problem, is that we wanted to work with a whole lot of taxa. And at the time, there was not a good way to take a lot of different organisms and put their DNA in a DNA sequencer at the same time, sequence it all, and then get it back out the other end and separate the data into the organisms that it came from. So I came up with a programmatic way of designing some tags that allow us to combine data together from many individuals, stick it into the sequencer, sequence it, and then basically demultiplex it or split it back out into the component parts on the other side. Then once we had our data in hand, I had to figure out how to put it all together. So I had to write a bunch of code and do a bunch of tests to take all of the data that we had and assemble it into a locus. Then the really hard part was that we could enrich the same locus from many taxa, but we had to figure out which ones were the same across all the taxa that we enriched. So we basically had to disentangle all of the data that we collected. And that took probably about 6 months of programming and screwing around with things to get it to work right. And then basically we had the data in hand. We did the same alignment and phylogenetic inference that we'd done before. And we found that we were actually able to reconstruct the known relationships between all these bird groups. Okay, so we collected data. We expected it to reconstruct these relationships. And it did with really high statistical support. And it gave us exactly what we expected. So now what I want us to do is I want to go from doing all of this lab work and computer work and tell you how I basically got back to working with real actual organisms again. So the first question that we wanted to work on was this question of turtles. So I'm assuming you've all seen a turtle. Turtles are really, really, really weird for a lot of reasons. One of the reasons that turtles are really weird is that we actually have no idea who the closest relatives of turtles are. So we don't know if it's birds and crocs and gators, or if their closest relatives are more like snakes and lizards. Okay, and this is a big, long, ongoing debate in biology. And one of the reasons that it's a debate is that morphological data, or sort of the data that you would collect from an actual turtle, seem to suggest that turtles are sometimes more closely related to lizards and snakes, and other times more closely related to crocs and birds. Now, molecular data say that turtles are sort of universally more related to crocodiles and birds. And there had been this study that came out, which I think now has largely been debunked, that said, oh no, all the previous molecular data were wrong, and turtles are in fact more like snakes and lizards. So you have all of these studies that were saying completely different things. And one of the things that we wondered was, well, can these loci that we found solve this problem, right? We get a whole bunch of them. They sample the genome pretty broadly, so we collect data from all over the genome, and we can collect a lot of data. So roughly 200 times more than we had ever been able to collect before. So basically what we found, after we collected all these data and we analyzed them, is that the data unambiguously said that turtles are more closely related to birds and crocs than they were to snakes and lizards. Just completely unambiguous. We had great results. They are definitely not related to snakes and lizards. And this result was later supported by 3 additional studies that sort of came out one right after the other. So one of the things that we also know is that fish are super confusing as well. So one of the things that we don't know about fish, which seems a little bit strange, is sort of which fish lineage came first. So who were the earliest diverging teleost fishes? Now the fish people, is anybody a fish person? I mean, other than liking to fish, does anyone work on fish in here? No? Does anybody like to fish? 4 people. So fish are the thing that we didn't know is whether the osteoglossomorphs, the clopeomorphs, the protoconthopterygians, or the asteriophyzons, or the allopomorphs came first, right? Who are the most ancient lineage of fish? We had no idea who that was. And one of the reasons we didn't know is exactly the same reason we didn't know for the turtles. Some papers have come out and said, well, we have absolutely no idea where any of these things go. We collected data, we analyze it, and it doesn't make any sense, right? And some studies said, well, these guys, osteoglossomorphs, came first, and other studies said, no, these guys, allopomorphs, and this is a tarpon if you have seen a tarpon, came first. And again, what we wondered was, can these data that we have figured out how to collect, can they solve this problem, right? Again, we're collecting a lot of data. The data are collected throughout the genome, and we are able to analyze a lot more of the data than we ever have been able to before. And basically what we found when we did the analysis of all this data is that, true to the last study, the allopomorphs, or fish like tarpon, are the earliest diverging fish lineage. And this topology, or this phylogenetic tree that we recovered, was very similar to a study that came out just a tiny bit before ours by a guy named Tom Muir. So one of the things that we've been able to do since then is we've moved beyond vertebrate taxa, and we've actually used this technique to try and understand relationships in Hymenoptera. So Hymenoptera are bees, ants. We figured out how to use this technique with museum specimens all the way from roughly the 1880s to the present. And primarily we've been using birds. And the nice thing about these birds is that we can use a very small part of their toe when they're in a collection like this, so we don't have to destroy the rest of the bird. And finally, we figured out a way to answer questions at sort of the population and individual level using birds, and we're expanding this work to move into other types of animals beyond birds. And if you're a fish person, or if you like to fish, one of the things that we've recently done is that there's been this really long-standing question in fish phylogenetics that has to do with the relationships of the spiny fin fishes, which are, again, a horrible name, the acanthomorphs, right? And these include things like billfish and what you can see on here, puffers, pufferfish, triggerfish, trying to pick things that you guys might know, snappers, etc. So all of these fish, we have basically no idea how they're all related to each other. And it's a ton of species. It's something like 23 or 25,000 species, and we don't know where any of them go. And we've recently used the data that we collected to, I think, answer a lot of these questions. Okay. So I'm almost done, but just to sum up, what I was basically able to do was to go from being completely frustrated about how to open the terminal on a computer, to learning how to program reasonably well, to learning how to use different operating systems, to taking all of that and integrating it and being able to answer a question that mattered to me. And just to reiterate, because I think it's important because people get frustrated when they do this type of work, this progress really comes incrementally, right? I mean, it took me a long time to get to where I even understand mostly what's going on. And I say mostly because there's still some days that it just doesn't make any damn sense. And again, with practice and a lot of work, you'll get better. But one of the things that's going to happen is you're going to be really confused a lot. You're not going to know what's going on. I mean, I'm confused every day. It doesn't bother me at all now. I've gotten used to it. And one of the things that you really need to do when you run into these problems is try and solve the problem yourself, right? You don't necessarily get anywhere when you're trying to learn this stuff by just relying on someone else's answer. It kind of takes the struggle of getting through the problem to understand what was going on. And for those of you who are interested in doing computational work, biology is increasingly computational. And it's, I would almost say, undeniably computational these days. The days of going out into the field and just collecting things and having that be it are probably numbered. And the other thing is that we can collect so much data now in a very short period of time that we have to use computers to help us analyze it. So in terms of sequence data, 1/16 of a current sequencing run produces, if you printed it all out in 12-point monospaced font and you print it on both sides of a piece of paper, a stack of paper 1,314 feet tall, okay? So this is a lot of data, and this 1/16 of a sequencing run takes about 3 days now to do. So you can make all this data in 3 days, and you have to make sense of it. And the only way to do that is computational. And if you're a computer scientist, I think that biology actually has things to offer you. If you're interested in algorithms or artificial intelligence, or you're interested in systems engineering and software engineering, we would love to have as many computer scientists come and help us in biology as possible. If you're a biologist, you probably need to start paying attention to computer science and maybe learn something about these algorithms and systems and software engineering. It's the future, and whether you like it or not, you probably need to know a little bit about it. And finally, if you want a job, right? I mean, usually most people want to get a job eventually. There are tons of jobs available for people who have some computational skill. It doesn't just have to be in bioinformatics and computational biology. If you are good at programming, if you're good at writing code, you can get a very good job in a very nice place pretty easily when jobs aren't all that easy to come by. Okay, so I just want to thank the other folks that work with me on the UCE work I described. There's Travis Glenn, Mike Harvey, who is a grad student here, and my buddy Mike Alfaro, John McCormick, Rob Brumfield, who is the director of the museum here, Laurie Sorensen, who is a postdoc at the museum now, although she was at UCLA when I was there, and then Brian Smith, who used to be a postdoc here, who is at American Museum of Natural History. And these are all the people that help me do all of my work. My funding's from the National Science Foundation and also from, strangely enough, a computer company. And I think that that's it. And if you guys have any questions, I'd be happy to answer them. Nobody? Yeah. Okay, cool. Yeah, we just use Illumina for the most part. The other ones, they're okay. We've tried them, but they're just not. Yeah, Illumina, it's 75% of the sequencing market right now, so that's pretty much what we use. Oh, okay. Yeah, so the data, yeah. Oh, sure. So the question is, when we collect the data, what do we do to basically clean it up? So when you collect these sequence data, I sort of glossed over a lot of detail. But when you collect the sequence data, you get nucleotides. So you get A, C's, G's, and T's in a sequence. And the machine gives you a quality score that goes along with that. And the quality score indicates your confidence in whether that A, C, G, or T is not only the correct nucleotide, that the order that it comes in is correct. Okay? So what we do is we use those quality scores that are estimated by the machine, and they're estimates of accuracy, to just trim our sequences. If you want to know the actual program you use, I use Trim-O-Matic. Yeah, and we just trim off the supposed bad parts. That doesn't mean that what we're left over with is 100% correct. It just means that we have some level of certainty that it's likely to be correct.
I know that in sometimes there will be some substitution error because of the poor calculation. Will that become a problem or not?
It is sometimes for very old samples. For the most part, when you have younger samples, because the reads that we have are random, they overlap each other. So even if you have an error in one nucleotide in this one read, we usually have many other reads that help us say, oh, no, that's not a C. We get a G 90% of the time. Okay, and it kind of comes out in the wash. Now, that's not entirely true all the time, but it's largely true most of the time. Yeah, sure.
I just want to ask, like you make the kind of registry, so you can program like some use maximum likelihood, some use capability, so how do you compare which one is more convincing?
That's a hard question. Okay, so the question is, when we generate a phylogenetic tree, what method do we use, and how do we know that that's the right method? Sometimes we don't. So we use mostly maximum likelihood. Sometimes we use Bayesian inference. When we use maximum likelihood, the reason that we largely use it is because it's the only thing that will handle the amount of data that we're giving it. That's largely the reason. The Bayesian programs run forever, and they don't tend to work well, and we don't get convergence in the data. Anybody else? Jake?
change it to do you were able to solve some problem that we had solved.
How trivial of a problem. Well, here's a good example. So a lot of things that you need to do are just cleaning up data, right? So you have a spreadsheet and some Yahoo has gone through and they have typed yes in capital letters sometimes and yes in lowercase letters sometimes, and sometimes a big Y and a little ES, and sometimes a little Y and a big ES, and you need to clean that up. Probably a month. But to get to where I can sit down and write computer code and not have to check it every 5 seconds to make sure that it's actually doing what I think it's doing, 15 years. Right? But it's not like I do it every day. It's a hobby, right? It's like the crossword. And now I can sit down and I don't have any problem with it. But it used to, it was a struggle. And you can't figure out, the most frustrating thing is when you do something, and the computer is almost always inherently logical. It does exactly what you tell it to do. You just tell it to do the wrong thing. And sometimes figuring that out doesn't make any sense. And those are the stumpers. But it takes practice. It's like anything else. But if you practice every day, it's just like learning a language, right? If you take Spanish here and you just go to Spanish class, it's going to take you a long time to be fluent in Spanish. But if you go to any Spanish-speaking country and you're thrown down in the middle of it and you don't have someone there to translate for you, you're going to learn Spanish pretty damn fast. So it's just a matter of degrees. And I would not even say that I'm really all that good at it. I know enough to get myself in trouble. Maybe a little more than that, but not that much. It's not what I do for my only job. All done? Cool. Thanks for coming, guys.